Okay, we're going to get going just a moment. Still have a few people joining us. Rakan, do you want to go over sort of the
Sure, the run of the
preliminary
show.
stuff? Yeah.
Yes. Hi, everyone. Welcome to today's webinar, Blueprint Neurotherapeutics on Small Molecules by Dr. Charles Cywin. So today he will have a presentation to present. There is a question and answer option that is available that should be available at the bottom of your screen. So you can go ahead and post all of your questions there, and Dr. Cywin will address the questions at the end of the presentation. Also, we will be sending a PDF copy of the slides after the meeting as well to everyone that is an attendee. And I'll pass it over to you, Chuck.
Great, thank you very much. Guess we'll just give it 1 minute and then I'll get going. All right. I've got my screen in order now, so I won't have anything popping up on y'all. Well, my name is Charles Cywin. I am the Program Director for the Blueprint Neurotherapeutics Network. And this webinar is going to tell you about our program, as well as give you some information briefly on a couple of other programs that are either feeder programs or related to it at the end as well, just so you have the little bit of bigger picture of it. Thanks. So the program itself combines the strengths of NIH and industry together. So, it's you all as NIH investigators and your ideas for novel drugs and drug targets for, developing strong assays and models and chemical starting points, combined with industry expertise through a network of subject matter expert consultants that NIH has hired to help you on these project teams once we get going, as well as contract services through our CROs that we have available. The overall goal of this program is to de-risk potential therapeutics to the point where industry will invest in them and allow for, hopefully the drugs to reach patients. You need somebody with deep pockets to pay for those phase 2 and 3 clinical trials and the marketing required to move a drug forward. And so we try to get enough of sort of that value inflection point in your program so that you can attract investors and get it to take it further. We hope to identify the best ideas for translation and through this funding mechanism and the associated infrastructure, move stuff forward. What should be noticed is it's really important for many people. This is non-dilutive funding. NIH is not seeking a stake in your company or your future things. And this non-dilutive funding provides access to resources that are not typically available in a lot of small businesses, concerns, or academic institutions. There are some that have it, and we'll talk about customizing your team. But the overall goal is really to preserve your intellectual property to facilitate licensing. And I'll talk a bit about it as we go forward, but the contracts and everything else are set up so that, and the program is set up so that your intellectual property stays with you. The program, as I said, is for academics and small businesses. It's only for small molecules. There are companion FOAs I'll talk about at the end for biologics. The projects can enter into either the discovery stage, I have just a hit and some assays, all the way to I have my clinical candidate identified. And so that is the point where you can do this. Hang on just 1 second. All right, and so the ability to provide these funds and this customized thing program for you is what really makes this unique at NIH. And the entry point along the spectrum is fine, but obviously the risk associated with the projects we should be de-risking it as we're going along. So if you're coming in as a development stage and saying as I'm a development candidate, the expectations for the milestones you've already met will be higher. And I'll talk about milestones in later in the presentation. So again, how do we set up our project teams? What we say we're going to do is set up a lead discovery team or lead development team that consists of you all as a principal investigator, our consultants, and our staff. And our staff all have drug discovery and development experience. Many of us come from industry backgrounds, doing different disciplines and having led teams in the past in industry. We are here to facilitate as NIH staff. We're not here to dictate what you do. Ultimately, this is your project, and you will be the overall driver of the science for these programs. But we're here to help guide you, keep on the critical path, make sure milestones are being met, and that we can continue the program. So what kind of contracts do we have? Well, we have contracts for medicinal chemistry, and that medicinal chemistry contract includes medicinal chemistry synthesis, as well as computational chemistry, as well as in vitro ADME characterization of the molecules. We have a PK and a tox contract that allows us to do in vivo PK studies, things like rat and dog, and rodent and mice, and as well as the toxicology required, so the dose range finding and GLP tox requirements to move a project towards IND. We have drug manufacturing formulation, so the ability to scale up material and do GMP synthesis, good manufacturing process to get material for the clinic. And formulation, typically we're aiming for, if it's an oral drug, a simple formulation. If of an IV, then it becomes a little more, sterile finish type activities. Ultimately, we want to go to the clinic, and we have a couple clinical contractors that can do first-in-human studies in phase 1. If you're doing SAR studies with us, then we have a relational database system, the CDD, Collaborative Drug Discovery Database, that we'll make available for bringing your data that you're generating with our data that we're generating from AMRI together with the chemistry all into one place. And again, that's useful for licensing down the road. But let's just say you have pieces already and you don't need them. You don't have to take it all. You could take some of it and not other parts of it. And we'll customize the team. But this is a very interactive team. It is a cooperative agreement. We do meet every 2 weeks and discuss progress. So it's very much like having a pharma team with you. We're with you every step of the way, all the way to the end. There are the limits on the projects we can take. They have to fall into the mission of one of these ICs that are signed up for this funding opportunity announcement. And they include NINDS, Aging Institute, Alcohol Abuse and Alcoholism, the Center for Complementary Integrative Health, Mental Health, Child Health and Human Development, the Dental Institute, the Eye Institute, and also the Drug Abuse Institute. So I think if you are in that space, it doesn't also have to, it's neurotherapeutics, so it's not just CNS, it's neurotherapeutics. So some peripheral targets are already being pursued within our program. We've already talked about having, a strong entry within the mission, entering anywhere along the spectrum. The exit point can also be anywhere along the spectrum, right? We hope to exit to external funding partnerships, and we've had people exit as early as lead optimization and have the whole program bought by a pharma, as well as all the way at the end after phase 1 and taken on into the further down the pipeline into, right, we have a couple of projects that are entering phase 3 after they've left us now. So, so either going to exit to external funding, to another grant, and of course, if we don't make milestones, there's attrition in the program. And so there are some times where the science just, the original hypothesis isn't borne out by the facts that when we do it, or we can't get the on scale up and repeating it, that the SAR doesn't repeat, or we get orthogonal SAR and we just can't move forward. So we will potentially end a program if we don't make the milestones. I should say that we don't do both primary and backup series. So we do that. We do multiple series in the beginning, but when we get to lead optimization, we're really focused on the primary series and getting that to a clinical candidate. A backup program would be something for another funding opportunity. These are phased grant mechanisms, meaning all grants must come into either the UG3 or the U44 phase 1 if you're an SBIR applicant. In that first year, that's sort of the kicking the tires of the program and making sure that things are where we were supposed to be. So, did the assays actually perform? Do the chemistry, does the compounds have SAR like they're supposed to? Do we have, a plan to go forward? It's really all generated based off of the milestones. It could be if you're in a development project, do you have a therapeutic index? If we do the dose range finding in the efficacy study and we don't have a reasonable therapeutic index, that could be a no-go for a development compound. So the early phase is about that. Then the later phase is, is an administrative review. We move forward to the U43 and then move into the later stages. So it supports up to a year in MedChem in the early phase and potentially up to 2 years, so a total of 3 years of MedChem. Obviously, the level of effort associated with that is not the same. In the early stage, we have a less level of effort than we have in the hit to lead and lead optimization phases. We do support all the way up through IND enabling studies and into first in man trials. Again, if you're going to use our contracts, and we'll talk about this, then how you designate this as either a clinical trial grant or a non-clinical trial grant is strictly dictated on whether you are actually performing the clinical trial yourself as part of your grant budget. Our contracts are not part of your grant budget. So therefore, if you're doing the clinical trial under our contracts, it's not a clinical trial grant, and you don't have to deal with the human subjects. That's because we deal with it on the contracting side. So as I said, all projects begin at the preparatory phase, and you can come in anywhere on the paradigm, but I should also point out that not all ICs accept development projects. For instance, NIDA and NIMH have language in there in the forward that says, we're not going to take a project at the development phase. We'll only take it at the discovery phase. And NIDA has great mechanisms for development projects that I think they would prefer you use. So in general, we come in, I said there's sort of a reduced budget. If you're coming the UG3, UH3 route, then if you exceed 500K in direct cost in any year of the grant, you must ask permission in advance. You must give me a detailed budget for all the years of the grants and ask permission in advance. And that process can take 6 to 8 weeks, which means you need to do that sort of right away if you've got a plan to be over 500K in direct costs in any of the years of the grant on this side. If you're on the SBIR side, that rule doesn't apply, but other rules on total costs do apply under guidance for SBIR. Our next application due date is August 10th. If you are a foreign applicant, everything is the same. You would come under the UG3, UH3 mechanism because that was the only thing you could apply under. And there's an extra level of review at the end that basically says, the reviewers will assess whether the project presents special opportunities for furthering research programs through the use of unusual talent, resources, populations, environmental conditions that exist in the other countries and are either not readily available in the U.S. or augment existing U.S. resources. So you have to make the case, and it can't be because we have an existing collaboration we want to just continue. You've got to make the case for why this really is unique and offers that unique opportunity. As I said, these are all milestone-driven programs, and it sort of gives you a timeline across the thing. Exploratory projects are really all coming in their initial milestone. That first year go, no go decision point will probably be around the assays, having a finalized target product profile and demonstrating, recapitulating when you benchmark that you have what you say you have for SAR, and can develop some early SAR on the molecules. If you're coming in hit to lead phase, well, that first year is going to really be about, do I have in vivo pharmacology that I can demonstrate after the end of the year by any route of administration, and has the chemistry advanced farther so that I can hopefully focus in on the series. If you're coming in lead optimization, well, the end of that first year would be all about picking your candidate, your clinical candidate. So do you have the efficacy, the reproducible, robust efficacy model data completed? And do you have, we would minimally do a rodent DRF to show that we have a therapeutic index done there. So a lot of people say, oh, I'm at the development stage, but I really haven't completed this. And so when I see people come in, I say, please consider coming into lead optimization phase and having these as your first year milestone to get things done, because it's going to take a while to scale your material up, to demonstrate the DRF and everything in that first year. If you truly have all of that data done and you already know where you're at, then you'd be in our pre-development phase where you could do large animal like dog DRF studies. We start to worry about having a tractable economic synthetic route available, having salt screen polymorph work done, having secured our clinical specialists in place so that we can plan for the clinical trial protocols. If all of that's done and you're really ready, then you would be ready for the GLP tox and moving in. If you're at this phase, please call me, because projects coming at this phase have a high hurdle for data, including a whole bunch of FDA required in vitro ADME data that you may or may not have. And if you don't have it, we'll want to generate that as part of your go-no-go decision point in the beginning. We do not take projects that everything is done, IND is open, and you just want to go to a clinical trial. Those kind of projects have another funding mechanism. So if you're just coming in for this late stage, obviously you can't do that. These two stages are typically done in the UH3 or the U44 phase 2, just so you know, and there are no direct mechanisms to do that. So you, generally speaking, you should talk to me about these programs if you're doing that. As I said early on, we do not seek to have a stake in your IP. Inventorship is, of course, determined by US patent law. So our program staff are here to facilitate you, but I'm a med chemist. I've worked for years in making molecules, but I'm not going to tell you how to make, which molecules to make, or interject into your project team at that level. I will say on a broader level that, are you considering issues that you should potentially be considering? But you'll have a med chem consultant that will work with you, and they'll have already assigned their IP to you, and they can guide you on these things. So the IP agreements with our consultants are put in place before they start to work with you, and they should aim for unencumbered IP. Likewise, if you have a multi-PI grant for multiple institutions, same thing. We want to know that you have IP agreements in place at the time of award, and that in your IP strategy plan that you are explaining that to the reviewers as well. Because the ultimate goal here is to create a licensable product at the end. There's a lot of information on our website. Here's our website listed. This is what it looks like. I can only fit so much on the screen, but below are all the program staff, including my name and email address here. All these links on the left, if you're including the funding opportunity announcements, frequently asked questions, other resources are certainly available here for you. So let me tell you also about some of the early phase. So let's say you're not ready for the BPN quite yet. You need to develop your assay and run a screen. Our NINDS Ignite program can help you with that if you're within the NINDS mission. You can also come to NINDS for things like early medchem optimization to just get to the in vivo efficacy studies to bolster and get you the preliminary data you need to move the project forward. You can also come in and to Ignite for things like developing an animal model in there. They have due dates coming up next week, and in October, Rebecca Roof is the program director for this program. And as I said, it's really designed to get you the preliminary data you need for a more robust application for BPN or BPN bio. It's an R61, R33 mechanism, and So it's a staged mechanism, but it's not a cooperative agreement. So you come up with, you have to write a clear milestone, and you're evaluated and done administrative review. If you meet your milestone, you get to move to the next stage. So clearly some things you want to do. So you want to read our funding opportunities announcements carefully. It calls for certain things to be included, such as including a table that says exactly, who's going to do what assays you need run, what the throughput of those assays are, where they're going to be run. If you're asking BPN to run things for you, that way we're sure that we can actually do it for you, and that what you're asking for is something that our CROs can provide. Our CROs cannot provide any disease biology support, but we can do the drug discovery stuff, as I mentioned to you. Stick to the page limits. Don't try to cram. Put forth solid preliminary data and discuss rigor associated with your preliminary data. Address the obvious criticisms. So if there's, a structural alert in your molecule, you should point it out and say, I have a structural alert molecule. I recognize it's there. And that's going to be one of the key SAR things we try to get rid of. In the absence of addressing it, people tend to ding you much worse on it. That can be part of your milestone, getting rid of X, Y, Z so that we can move forward. Clearly indicate, as I said, what's going to be done, and then it calls for a table to do that. Also, and it happens every round, I get an application or two that people submit budgets for each phase rather than every year of the grant. So every year of the grant you must have a budget for. So if it's a 5-year grant, there should be 5 individual grant year budgets that I should see, not just one big UG3 and one UH3, or one U44 phase 1 and one U44 phase 2. So be careful. Give me all the budget years. Like I said, we talked about, discussing the rigor, talk about the controls being used, it's being blinded, is it properly powered, how you're eliminating the bias in these studies is really important. And you do that in the research strategy section. Don't bury it in your vertebrate animal section. You need to, really discuss it succinctly, but discuss it. Complete all your registrations at least 6 to 8 weeks in advance, which means if you haven't started your registrations, start today. If you're going to come to the August receipt date. Consider submitting your application at least a few days early. That will give a chance to react to issues that might come up. So if I see something wrong with your application, I can contact you, and I try to look at these applications as they come in. But if you submitted on the last day, I'm obviously you're not gonna have time to react to anything I have. If you're applying, as I said, to the UG3H3, any year the grant goes up 500K, you need to make sure that you request permission to come in and provide me detailed budget application. Wakanda, can I ask you to stop pasting in, because I'm getting a lot of beeping in my ear. Can you make sure you talk with your tech transfer and BD group about your need for licensing and patents. It's important to plan ahead. We can't support the cost of patents under the application, but we can and do recommend you have something in your application in sort of that year 5 that deals with the filing of the IND, because you'll need to have a regulatory consultant. We provide regulatory consultants up to the point where to do. Please don't make this your only source of funding in your lab, because this milestone driven can end abruptly. Don't under-resource your budget to avoid the limits. If you really have to be above 500K, talk to me about it. Sometimes you can get away with, like I said, a lot of you are using our contracts, and oftentimes you're not really pushed to be near that 500K. But there are times where it's required, and you should talk to us if it is. Don't plan to use our contracts for disease biology. I mentioned that already. Or one-off in vitro assays. So if you're coming in as a development project and say, well, I just need to get a HERG assay done, contract it yourself, because it's a lot of work to set up a government contract for one experiment. It's not worth it. It'd be much easier, faster for you to do it yourself. Do not code this application as a clinical trial if you plan to use the BPN contractors. I can't point it out this enough. You don't have human subjects, you don't have clinical trials. If that the only thing was the phase 1 being done in our contract. If you do plan to do it, consider whether it's a delayed onset clinical trial, if it meets those rules or not, because you may or may not know what you're doing well enough to have a protocol written at an early stage of the program. We had the August receipt date. The PAR 20-122 is for all applicants, including small businesses that may or may not be eligible for SBIR, due to ownership, due to other things. And any SBIR eligible small business is encouraged to apply to the PAR 20-111. All applicants are reviewed by the same special emphasis panel. We have things with a lot of the indications listed here, but look in the FOA for what each IC is presently interested in. And I promise I touch on a couple programs at the end here. And so the first one is our newly launched Blueprint Neurotherapeutics Network for biologics. So much like what you just saw for this, we have set up a program, and the first receipt date is in August 10th for this program to do biologics and things like peptides and proteins, novel nucleotides, gene therapy, cell therapy, other emerging biotechnologies, all will fit under this. Anne-Marie Brome is the program director for that, and I encourage you to look at the FOAs listed here if you have a biologic, because we now have a home for you. There's also, if you're in the pain-related area, we have the non-addictive analgesic therapeutics development program, and this also does small molecules and biologics. It is mirrored after the Blueprint program, but also has a requirement for that you consider a companion target engagement biomarker. And it can potentially go a little bit further we're trying to get it all the way ready for EpicNet. So it does have resources. So if you're in the pain area, you potentially have 2 different funding opportunity announcements you could consider. The receipt date for this one is July 14th, the next one up. And this is an RFA, so this has set-aside money associated with it, where the BPN does not. So that being said, I'm here to answer questions about the application process, the review process, proposals and budgets, the contracts, intellectual property, all these topics or anything else on your mind. And at the end, you can ask the questions. I'm going to start fielding those now. But if I don't answer a question or don't get to it, you can always email me. We can do that in the future. So Rakonda, let me go ahead and open up the thing. I'm going to pause my screen. And I'm going to see if I can get this open. Okay. As I move this up here where I can see it all. Bear with me 1 second. All right, so questions. Can you talk about eligibility or foreign entities? So I did touch on that already. If you are a foreign institution, you would come under the UG3, UH3. Or even if you're a foreign component of the thing, you need to come in on the UG3, UH3 because SBR doesn't allow foreign components for the most part, unless you can really justify them. But no foreign entities could be under SBIR. So next question up is, my company has submitted an SBIR application to NINDS. Can we also submit to the Blueprint Neurotherapeutics Network? So I guess the answer to that is it depends. You can, if you've submitted an application to NIH for anything, anywhere, you can't have 2 applications under review at the same time for the same aims. That's not allowed, no matter where it is, whether it's SBIR or non-SBIR. So, but once you get your summary statement back, you are free to apply to a different funding opportunity announcement if you want. So if you have already gotten your funding up, already gotten your summary statement back, you can reapply to the SBIR program, you can apply to the BPN, but you can't apply to two things at once. And whichever one came in first is the one that's going to be considered having priority. STTRs we do not have an STTR version of this program under this FOA, but under the UG3 UH3 mechanism, you can certainly have a collaboration between a small business and a academic institution. And if you can maintain the relationship under the SBIR rules, you can have an academic relationship, but obviously the money split is quite different from STTR in that scenario. Are foreign entities eligible to apply to the R61, R33 for early medchem to get more robust data? I believe so. Same kind of rules would apply in terms of showing the uniqueness of it. And we'll go from there. Our lab has a compound which has a new sedative anesthetic, which can also use a potent anticonvulsant as well in treatment of acute alcohol and drug withdrawal, which would be best mechanism for the application in BPN. So this is a common question I get, and my answer back to people is, think about it from a couple different perspectives. I can't give you a solid answer on this one, but the way I advise people to think about it is, first, where is my robust data? Where is it I'm going to have the data that supports the indication best? Because that's where the reviewer is going to look from a scientific point of view and say, this is reasonable. And if it had there, it would have an impact. But there's also the commercialization aspects of it. It's not uncommon for, a project to say, want to go to one disease that has a big market, but the path there is through a smaller market because that's the most straightforward proof of concept clinical trials. If that's the case, you may want to go that way because scientifically it's easier to grasp and it'll be easier to get commercialization for that. But ultimately, wherever you go, that institute that's funding you wants to know that you're staying on their mission throughout the process. And the other things are, are expansion. If in fact you are going to change, then you could end up losing the funding because you change. And I've have companies come in and say, pivot from a small business. They got new management and say, well, we're not going to do that indication area. We're going to do this indication now. And unfortunately, it's being funded by one institute and they don't want to fund for that indication. So the funding stops at that point. Especially if it wasn't reviewed for that indication by the reviewers. But if you have multiple indications and you can say in the grant, that's what we plan to do. We plan to, we ultimately have a portfolio of indications, but we're going to focus on this one first. And we may do other animal model work through other funding to further bolster these other indications on the outside. For the IGNITE program involving establishing better in vivo efficacy, which animals are available? That is something you're doing yourself. So I think it's pretty much whatever you propose. I think you could potentially propose dogs and primates. Can you do it within the budget? Because they're somewhat limited on budget on those. I think it's like up to $750,000 across the entire 3-year period. So you just have to see whether you can actually do it within that budget. If our proposal involves BPN contract resources, how do we get information as to their capabilities as to incorporate such in our grant application? You can talk to BPN staff. You don't have to spend a lot of time discussing the capabilities because the reviewers are instructed to assume that all the NIH CROs are being done to industry standard. And so they're not subject to review. So what you really need to find out from us is what you're asking for. Can we do it? If we say we can do it, then you can just state we plan to do this through the NIH CRO, and then it's really no longer up for review. We have a hit to lead that is that's efficacious, identified, and established using in vitro and vivo testing and transgenic AD model. However, we feel there are better candidates covering a wider, stronger patentable space. We've already made 10 molecules exploring the new chemical space We want to take them through the same in vitro and vivo testing funnel that got us the original hit compound. Can this form the basis of our new application? It could. You could say, I have this as a lead and proof of concept, but it has these issues, and we have these other hits, then that's going to be the basis of our early stage program and what the milestones are to sort of focus in. At that stage, you're probably being a hit to lead, and you're really trying to establish sort of the series in that mode. Is the small business applicant allowed to be employed also by academic institution to apply to your awards? Or must be 51% by the small business. It's the same SBIR rules as every other SBIR rule. So I believe the 51% thing requires. But if you have a question, I would talk to the SBIR person from whichever IC you're interested in from a disease point of view to be certain. Is funding for 3 years from NIH approval or patent filing dates? I'm not sure what that question means, or whether it's referring to the BPN, which doesn't necessarily have a 3-year date, and that we don't fund patent filings per se. So whoever stated that question, could you be a little more specific? Let me see. If I'm applying from a company, then this is the only SBR funding. There is no SCTR-like mechanisms. I've answered that question correct. And if you have a strong phenotypic response, mode of action is not completely clear, can we propose a mode of action alongside studies to identify the lead molecule? Basic research into mechanism is not in scope. However, you can apply for an R01 to do that kind of work as a supplement to the BPN. So there are, or an R21 to do that kind of work, as well as the BPN to do the actual SAR work. How should a letter of intent be submitted 30 days? So you want to submit a letter of intent to me as the program director as early as you can, and understand it's not an absolute requirement to put a letter of intent, but the reason why we ask for a letter of intent as early as possible is those go to the review staff, and the review staff can start recruiting the correct reviewers for your application. So by giving us as much notice as possible, it improves the quality of the reviewers and their reviewers' expertise relative to your application. Is brain cancer a combination of therapy accepted? Brain cancer is within the mission of NINDS. Combination therapies you should talk to me about, because oftentimes that's a little more complicated and may or may not be. Do you consider the clinical development of synthetic peptides that are not biologics as defined by FDA? So where the blueprint for small molecules, BPN, we sort of say 6 mers and lower will handle. Anything above that should go to the BPN biologics group. But if it's peptides in general, you can also go to the BPN biologics group straight out of the gate, so that you actually have a choice there. Can the total budget exceed 3 million? I assume the answer to that question, I guess with permission, it could. If it's an SBIR, again, you sit within the limits of what the SBIR rules, but yes, you should budget what you think you need. We'll talk about your budget, but if you need to come in more than 500K, make sure that you do that early if you're in the UG3, UH3 mechanism. And is TBI a potential application? Yeah, traumatic brain injury, again, falls within NINDS's mission and can certainly fall. Now, there's something about a July 5th only application date. So the application for BPN is actually August 10th. I'm not sure what the July 5th date is referring to here. And the regular application deadline is October 5th. Again, I'm not sure what you're referring to. The only application for BPN is August 10th and then February of next year. So it comes out every 6 months. Would you consider supporting a CNS drug delivery platform technology for small molecules? If it's just developed the platform technology, the answer is no. This is all about developing a drug molecule. But there are other mechanisms you can talk to us about, and NCATs and other places may be appropriate for developing a platform. Would BPN consider a peptomimetic for amino acid peptide? Yes. and it would fall under small molecules in those cases. We would like to identify novel compounds based on a new hypothesis, effectiveness of a known drug, I think there's a known drug in vivo receptor target. Identify novel compounds based on a new hypothesis. Again, it could. The key elements here are, am I developing novel intellectual property around the compound so that I'm going to have control over it and can license it at the end and take it and commercialize it so it gets to patients. I answered the next question up was asking about the next submission date after August 10th, and that is in February of 2022. All the receipt dates are listed in the funding opportunity announcement. We have next question up is we have 2 molecules that are bioavailable, have efficacy, but we have 20 backup molecules on different scaffolds. Can we write an application to characterize the backup molecules? In theory, you could, but again, we're all trying to get to one molecule, and it becomes a little bit of a sticky thing because when you try to do this, people are going to ask what's wrong with the original molecules that you're trying to fix, and sometimes you run into issues of, well, let's see the results of those first molecules before we spend more money on the backup molecules. So you have to make a real compelling case when you want to do that. But we have done backup programs for instance, we've had people who say, I have an IV formulation for this lead compound. We want to now develop an oral formulation for a different compound for a different indication. And we've done that. For the IGNITE program, can a small business applicant be employed? Okay, so it's not an SBIR program, so IGNITE would fall under, and I suggest you talk to Rebecca Roof about the eligibility requirements and look at the funding opportunity, but it's not an SBIR rule. It's not SBIR, so the SBIR rules don't apply. If the PI is awarded an R35 from NINDS, does the Blueprint remain completely separate from other NINDS grants are rolled in? It is not considered separate, but it is considered intrinsically expensive funding, and it would be subject to council review at the end. So hope that answers the question as best I can for you. Again, if you want to talk to me about specifics, I'm happy to talk to you more about it. So Rakonda, any other questions?
Hi, Chuck. That's all I have right now.
Okay. I went through those questions quickly. If there are any other questions, feel free to post them. As I said, we will be giving copies of these slides out to help. And we do, you know, if you're preparing for the application, you can reach out to set up a phone call with us in the intervening months here ahead, and I'll be happy to try to arrange time to talk with you. I get a lot of requests, and I typically ask for some information in advance, and if I can answer questions by email, I try to do that, but as quickly as possible. But I'm happy and do, Rakonda will attest, talk to people pretty much daily, and so it's, you know, it's an integral part of what I do. I'm here to help as much as I can.
Had a couple more
Sure.
come in.
Okay. How is it possible for me as a faculty member to participate in SBIR as a university-based contractor to do efficacy studies paid by the company's SBIR funding? You would just be a co-investigator on their application. In that case, it's not dictated by, you know, they can have you on there, provided a couple things are true, that you're a US-based institution, and that it doesn't somehow make them no longer eligible for SBIR funding because they have too much money going out the door. For instance, in a SBIR in phase 1, 66% of the money is going to stay with the company. That's not being used for fee for service or other kind of things like that. And then in phase 2, 51%. So if your work with them doesn't make them no longer SBIR eligible, they're welcome to do that with you. The other way is to come under the UG3, UH3, not the SBIR program, and then the SBIR rules no longer apply, and you can do funding the way you want. Rakanda, can I ask, make sure that you submit the questions from each individual person individually so that I know that one question is not related to the next question. So I got a question, can foreign CROs be used? So again, yes, for the UG3, UH3, you can have foreign components. Not really for the SBIR. You'd have to, it is possible, only after you demonstrate to the SBIR people and it's approved at the high levels of the SBIR in the office of the director, that there is no other choice but to use this foreign institution, and that sometimes they can do it. But if you can well justify it, SBIR will consider it, but it has to be very well justified. So we have a strong phenotypic response. I think I answered this one. Strong phenotypic response to a mode of action is not completely clear. Can we propose mode of action site? Yes, I did answer that question prior. The answer to that question is no. Mechanism, disease mechanism and things is not within scope of the BPN, but you could apply for an R21 or R01 companion kind of grant separate to do those kind of studies. And I have had BPN people working in phenotypic space have both an R01 grant to do these kind of work as well as the BPN to focus in on the actual drug discovery process. Well, again, I really want to thank everyone for taking the time to join us today. And if there are any other questions, feel free to email me. Always happy to answer them. And I wish you luck in applying to our program or other programs, and we're here to help make the connections for you, so feel free to reach out. And I guess if there's no further questions, I'll wrap up now. Kanda? Any closing things?
Nope, no further questions. Chuck, again, this will be sent to everyone, a PDF copy of the presentation as well as a recording of this. And as Chuck mentioned, you do have his email for any questions that may come after this is over.
All right. Thank you all again for taking the time. I really do appreciate it. And best of luck in writing your applications. Bye now.